Okay, so this is going to be the second part of the energy and enzymes PowerPoint. So the first part was all about thermodynamics and free energy and exergonic and endergonic reactions, meaning break the catabolic, exergonic, or building up, anabolic, and endergonic reactions. And ATP hydrolysis is one of the most important exergonic reactions that we have. So we use that energy from ATP hydrolysis to drive a lot of endergonic reactions. All right, so let me share my screen again. Oh no, I tried to join my living room again. Whoops, need to join the iPad. All right. Okay, so I hope you guys can't see that weirdness in the background. All right. Okay, so cells do three main kinds of work, right? They're really categories with three main things. The first is chemical. Cells have to do a lot of chemical work, meaning they need energy in order to make, chemical bonds, right? To drive these endergonic reactions of putting two reactants together to make a product, right? So again, the cell uses ATP to put, here we have two reactants and we're building up to create a product, right? And again, the building up is always going to be endergonic and anabolic and you're going to need energy to do that, right? The second main kind of work that a cell does is mechanical, right? And this is the energy to move proteins. For example, the example that we saw in that video was that crazy motor protein, like walking along the microtubule. It was took those like literal steps along the microtubule. Another example shown in this, shown down here in this image, is cells using ATP to drive a motor protein. In this case, it's not a microtubule, but this blue is actually actin or a microfilament, and that allows the filaments to come closer together, and that's how we move our muscles. All right, the third kind is transport. And we already talked a lot about this in chapter 7, where it is the energy to move particles across a membrane, right? And so, for example, this is really important for creating electrochemical gradients. And what I mean by that is if we have a membrane and we have a few particles out here and lots in here, meaning inside is a little hypertonic, then we can use ATP to continue to shove more blue molecules over here, right, against the concentration gradient. And that's going to be really important for creating these electrochemical gradients. So cells do three main kind of work: chemical, make bonds; mechanical, move things, move proteins; and 3, transport, moving things across the membrane. So enzymes are considered catabolic proteins. They can be defined as a protein that can speed up the rate of a chemical reaction. And what's really cool about enzymes is that they themselves do not go under they do not change. They have like superpowers. They can speed up reactions, they can do work, but they do not change, right? So again, an enzyme is a protein. It can speed up the rate of reaction, and the enzyme itself does not go through any change. So if we again look at our free energy diagram, we have free energy on the y-axis, the process of reaction on the x-axis. Almost all reactions start with reactants and products, right? In this case, we have reactants that have more energy than the products. So again, that is going to be a negative delta G because we are going downhill. But it turns out that most reactions actually have this large kind of hump before they go from reactants to products. That is called the activation energy. That is the amount of energy it takes for a reaction to go, right? This is the activation energy right here that I'm drawing here. So this is the activation energy. And it turns out that the way that enzymes work is that enzymes work by lowering the activation energy of a reaction. So, for example, I'm going to erase that. So here is the activation energy without an enzyme, plus the enzyme. We are going to see that that activation energy is lower. It was reduced. So enzymes, they speed up reactions, and they do that by reducing the activation energy, which is this large hump in a reaction. So enzymes, again, are proteins, and their three-dimensional shape allows them to be substrate specific, all right? In a substrate, all that is something that binds to the enzyme, all right? It's something that binds to the enzyme that the enzyme is going to do work on. So because of that three-dimensional shape that's super important, which we learned about in all those primary, secondary, tertiary, quaternary folds of a protein, during that three-dimensional shape, enzymes all have what are called active sites. And an active site is a region on an enzyme where a substrate molecule binds and therefore can undergo some chemical reaction, right? The reason I always draw it as a Pac-Man is that I always think of the enzyme as being able to eat or bind the substrate, right? So enzymes are substrate specific. They can only bind whatever substrates fit perfectly into their active site, right? So here we have our little purple enzyme. It has an active site, and substrate A cannot fit in, substrate B cannot fit in, substrate D cannot fit in. However, substrate C is a perfect fit, right? So only substrate C will fit perfectly into that active site of the enzyme. So this is the only substrate that will bind it. So enzymes have active sites where substrates will bind, and they are substrate specific. Only the correct fit will work. And this brings us to the model. So we've been talking a lot about different kinds of models, such as like the fluid mosaic model, and those kinds of things. So there's another model for enzymes, and it's called the Induced Fit Model, right? And this was not around when I was, I forget what year this came about. When I was first learning about this, we learned the enzyme and substrate as a lock and key. Like you put a key in and it unlocks it. But now we know through a lot of microscopy and other methods that the induced fit model is more accurate. So this states that as a substrate binds, the enzyme changes shape, leading to a tighter binding, which brings chemical groups into position to catalyze the reaction. So instead of a lock and a key, it's more like a handshake. So as the substrate fits in, the enzyme actually binds tighter, right? So they're both kind of changing shape to have a better fit. And I'll show you a picture of that. Why is my computer not responding? Come on. Just a minute here. I'm going to stop share and try again. So just a minute. Let me try that again. Okay, so this is, so we were just here and now we're here. So induced fit model. So the previous theory that is not accurate anymore, it was called the lock and key, which just stated that it was a perfect fit of enzyme to substrate and it fit in perfectly and nothing changed. We now know that an induced fit model is much more accurate. Instead of something just binding and fitting perfectly, we know that when substrates bind, the enzyme itself actually changes shape to bind more, to bind better, right? And you can see that in actual molecular data. So here's an enzyme called apohexokinase. When it binds its little substrate shown in red, the enzyme actually physically binds tighter. So again, it's more like a handshake where the enzyme is actually going to grip onto the substrate. So enzyme active site changes upon substrate binding, and that really is the induced fit model in simpler words. Enzyme active site changes shape upon substrate binding. All right, so enzymes have three super important properties. One, they're substrate specific, right? Each enzyme works with a specific substrate, depending on that chemical fit. The second is that enzymes are not consumed in a reaction. A single enzyme can catalyze thousands upon thousands of reactions per second, and they remain unaffected. So the enzyme at the beginning of the reaction is exactly the same as the enzyme at the end of reaction. They do not get used up. They do not get changed. They're superpowers. And then the third one is that the enzymes are affected by their cellular conditions. And we know this already because enzymes are proteins. So any condition that affects that protein structure, such as temperature, pH, different salinities or salt concentrations, will affect the enzyme function. So we know that the active site is dependent on its three-dimensional shape. And if that protein gets denatured in any way or loses its folds, then it will not have that active site anymore and it will not be able to work. So for example, this is a diagram that kind of shows how enzymes have optimal environments. So for example, if we look at pepsin, which is an enzyme in our stomach, its optimal pH is about 2.8, right? If you go farther from that pH, the pepsin still might have some activity, but if you get beyond 5 or 0, then the enzyme is not going to work at all. Similarly, if we have enzymes, for example, that are in our, so trypsin, for example, which is in our small intestine, the very top of our small intestine, the pH is much higher. So for example, its optimal pH would be at the top of this peak, which is about 8.6 or so. And that is where it's going to work the absolute best. As you go out farther than 11, it won't have any activity. If you are, let's say, pH 9.5, it'll work, but it'll work about half as well, right? Because it's partially denatured. So every enzyme has an optimal temperature. That's pH, temperature, salinity, etc. The rate of an enzyme-catalyzed reaction, so the rate of a reaction, depends on the concentration of the enzyme and the concentration of the substrate you're dealing with, right? So we're going to talk about this more in class, but it's really important when you're looking at these graphs to look at the axes and really try to work out why the graph looks like this. So let's start with this first graph. So here we have the concentration of enzyme on the x-axis and the rate of reaction on the y-axis. So as you increase the enzyme, you will increase the rate of the reaction, meaning you can have more substrates kind of reacting to give you more product. This second graph, students always have a hard time with this. So here we're looking at the rate of a reaction on the y-axis, and here we're talking about the substrate concentration, or the amount of substrate. Remember, the substrate is what will bind in the active site. So here, as you increase substrate concentration, the rate of reaction also goes up, but only to a certain point. And that is the point of saturation, right? Where the enzyme, if we have a certain amount of enzyme, so in this example, we have a limited amount of enzyme, right? The enzyme concentration is not increasing. We have a certain amount of enzyme. If you add some substrate, it'll go up, it'll go up, it'll go up until every single enzyme already has substrate bound to it, and you can't go any faster than that, right? So the point of saturation is when the active sites are all bound, and I have an image to show that. So again, for all of these graphs, you have to know what your axes are showing, right? So the reaction rate is on the y-axis. Here we're talking about the concentration of substrates. In this example, we have a total of 3 enzymes in this reaction, right? We're not increasing enzyme here, we're only increasing the amount of substrate. So we have a total of 3 enzymes, and at low substrate concentration, you're going as fast as you can. So these substrates here, the enzyme is moving as fast as it can, right? Because there's even more enzyme to go, so you can go faster. In the median substrate, you can see that 2 of the enzymes are full, but we can still get a little faster because that enzyme can bind as well. However, at high concentrations of substrate, every enzyme is occupied by a substrate, so you can't physically go any faster, right? Unless you add more enzyme, right? That's one way you could get faster. But if we have a limited amount of enzyme, increasing the substrate concentration is not going to help you. All right, so there's 2 things now that can help enzymes work better. The first is called coenzymes. These are non-protein. They're still organic molecules, but they're not proteins, right? And they bind to enzymes, and they help them work better. An example that we're going to see a lot is NAD and FAD. Another one that we're going to talk about later is called coenzyme A. So again, coenzyme is non-protein based, right? Whereas cofactors, the other kind, are also non-protein, and they're small inorganic compounds, usually ions. And this is magnesium, potassium, calcium, zinc, iron, copper bound. So coenzymes, non-proteins, and these are organic molecules. And remember, organic means carbon and hydrogen, whereas cofactors are usually ions or small things. And the example of this is hemoglobin, which is the oxygen carrying compound in our red blood cells. You can also have things that can inhibit enzymes, right? So these 2 increase the function of the enzyme. So cofactors and coenzymes, they work together. They help enzymes. But we also have things that can inhibit, all right? And they can do it in 2 different ways. The first is called competitive inhibitor. And this competes with binding for the active site, right? So in a normal reaction, we have our substrate here as a little green piece of pie, and we have our enzyme. This little red part that I'm drawing represents the active site where a substrate will bind. Again, all enzymes are substrate specific. In this reaction, the little blue enzyme, what its job is to do is to cut that piece of pizza into 2, right? That's what the enzyme, that's the work it's going to do. Here, in the competitive inhibitor, a competitive inhibitor will compete for the active site. Therefore, it will bind, and little green pie here cannot bind. What this means is that competitive inhibitors look like the substrate. They resemble the substrate because they are going to fit in that active site. So they have to look exactly like that. Non-competitive inhibitors, on the other hand, bind to an enzyme away from the active site. Any site away from the active site is called, so away from active site is called allosteric, right? Allo means other. So away from the active site, it'll bind at an allosteric site, right? And what this does is that it will bind somewhere away from the active site, and that's called non-competitive. It cannot compete at the same active site because it binds an allosteric site. All right, we'll talk about that in a second. All right, so enzyme control. I'm going to introduce these three and then we're going to talk about them each. All right, so allosteric, so these are ways that we can control enzymes. One, allosteric binding, right? When an enzyme's function is affected by the binding at a site away from the active site. So allosteric, again, away from active site, right? Feedback inhibition, this is when you have a metabolic pathway where we have enzyme, 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 going from substrate to a product at the very end of the line with many different enzymes. And it is when the metabolic pathway will then either turn off, it'll turn off when there's enough product. So basically it says enough product, stop those initial reactions. And then the third one is cooperativity, where enzymes of 2 or more subunits, if one substrate binds, then it'll trigger the other ones to do it. So allosteric regulation, when an enzyme's function is affected by the binding away from the active site, right? And this can come in 2 flavors. You can have something that reduces the enzyme function, and that's called allosteric inhibition. And then you can have allosteric activation, something that binds away from the active site and increases the enzyme's activity, right? So if we look at inhibition, so this is a form of non-competitive inhibition, right? Non-competitive inhibition, where you have your inhibitor binding away from the active site, and this just makes for a bad enzyme, for an overall bad enzyme, meaning you can't compete off anything. It just has now changed the shape of that active site, and it's just going to be bad. Allosteric activation, on the other hand, is a molecule, in this case an activator, that will bind away from the active site, and it will open the active site up perfectly so the substrate can bind. So we can have molecules bind away from the active site to either act as a non-competitive inhibitor, right, allosteric inhibition, or something that binds to an allosteric site away from the active site and makes a better enzyme, or makes the active site open. All right, so for that feedback inhibition, which is the second way that we can kind of regulate enzymes, this is an example of a metabolic pathway where we have 1 enzyme, 2 enzyme, and 3 enzymes all working kind of in a pathway. Here we have enzyme 1 turning green triangle into, I don't know, we change it into whatever, into a purple thing. So we have this chain of reactions, right? One enzyme gives a product, and then that product goes to enzyme 2, and then that product of that goes to enzyme 3. Feedback inhibition is when a metabolic pathway eventually gets turned off because the end product here is going to say, dude, we have enough, right? So the end product is, let's say if we're making, for example, I don't know, the buildup of glucose during photosynthesis or something. Like, dude, we have enough glucose. We don't need more. So what it's going to do is that end product is going to act as an allosteric inhibitor, and it's going to turn off one of those enzymes, usually at the very beginning, right? So a product at the end is going to go and turn off one of the enzymes at the beginning, saying we're going to turn off the whole pathway because we got enough. The last way that we can regulate enzymes is through a process called cooperativity. Here, for example, we have an enzyme that has 4 subunits. So we have 4 subunits that come together to make a single functional enzyme, right? One enzyme here. And what we see is that if you bind one substrate, that will then open up the sites so that we can bind all of them, right? So if one substrate binds, it causes an induced fit conformational change, which then can trigger all the other ones to bind as well. All right, so that's it. Remember, I have lots of questions. This stuff is not trivial. You've probably never heard of a lot of these things before, so please do not wait to study because this test is meaty. It's got a lot of stuff in it. It's got everything from all the parts of the cell to membranes and membrane transport, to enzymes and energy. All right, that's it. Signing off. Adios.